Okay, so AI is already making a huge impact on healthcare. But what about clinical trials? In fact, AI and machine learning tools are transforming how clinical development occurs. And advances in analytics technology will offer new opportunities that drive further benefits to the clinical research landscape. Hello, everyone. Today, we present to you the second installment of AIMS, Harnessing the Power of AI to Transform Clinical Trials, co-organized by the NEJM Group and JMed. This is Zhao, Deputy Editor of NEJM Frontiers in Medicine. So tonight, we have a packed schedule in the next 2 hours. First, we'll have 3 presentations, each followed by a 5-minute Q&A. We'll then have about 40 minutes roundtable discussion. So first, let me introduce our NEJM editors and some leading lights attending the event. And let me share my screen. Okay, so our forum is going to be moderated by Dr. Jeffrey Drazen. So Dr. Jeffrey Drazen is Editor-in-Chief of NEJM Evidence, a new NEJM Group journal launched in January 2022. He is also the NEJM Group editor. Previously, he served as Editor-in-Chief of the New England Journal of Medicine for 19 years. And Dr. Beth Hamel is Executive Editor of NEJM. She has a deep interest in clinical trial methodology. So I would pay close attention to Mary Beth's perspective in using AI in clinical trials today. And Dr. Zhao Kohane is an editorial board member of NEJM. Dr. Kohane is the chair of the Department of Biomedical Informatics at Harvard Medical School. He develops and applies computational techniques to address diseases at multiple scales. And Dr. Charlotte Haug is international correspondent of NEJM. Charlotte and I have been co-organizing the AIMS meetings since last year. So our current editor of Editor-in-Chief of NEJM, Dr. Eric Rubin, so due to some power outage at his house, he has to eavesdrop in the background. So we won't see him in person today. So let me first welcome Dr. Jeff Drazen to deliver the opening remarks.
Thank you, Jean-Fei, and good morning, good afternoon, good evening, or good night, everyone, depending on where you happen to be. And thank you for attending the second AIMS meeting. In the first AIMS meeting, we talked broadly about how artificial intelligence or machine learning could make a difference in the practice of medicine, in the way that information is gathered to help physicians make better decisions. In fact, the entire mission of the NEJM Group is to get to health professionals the information they need to do a better job taking care of their patients. So here early in the 2100s, the 2000s, how much information are we gathering using technology that was developed in the early 1900s? And how much information are we gathering that we are using the best technology possible at the current time? Today's AIMS meeting is going to focus on the gathering of information that is used by health professionals around the world to make decisions, the best decisions for their patients. And how can artificial intelligence, which is a way to help us do thinking that we couldn't actually do on our own with any reasonable reliability, and to use that thinking to improve the way that we gather the evidence that makes a difference in how patients are treated. So the question that we're asking today is, how can AI change clinical trials? The key way that we gather evidence to make a difference in medicine. And with that, Jean-Fei, I'll turn it back to you.
Thank you so much, Dr. Drazen. So we also invited several physicians or surgeons to share their insights in using AI to improve clinical trials. We also have some leading AI scientists to talk about this topic. So let me share my screen again. So let me briefly introduce each of the discussants or speakers. So we have Dr. Arnaub Chatterjee. He is Senior Vice President of Acorn AI, part of Metadata Solutions. In his spare time, he also is a teaching associate at the Department of Healthcare Policy at Harvard Medical School. And Dr. Adam Dunn is Associate Professor and Director of Biomedical Informatics and Digital Health at the University of Sydney. And for those audience who probably listened to our last year's AIMS meeting, Adam Dunn actually gave another talk, but he wasn't able to join the discussion. Today, he is going to answer the questions and join the discussion. And Dr. Junbo Ge is a member of Chinese Academy of Sciences. He is Director of Department of Cardiology and Interventional Cardiology at Zhongshan Hospital of Fudan University. And Professor Shengxian Tu is a professor at the School of Biomedical Engineering of Shanghai Jiao Tong University. And Dr. Qiang Zhao is professor and director of Department of Cardiovascular Surgery, and he is vice president of Region Hospital at Shanghai Jiao Tong University School of Medicine. And Dr. Yunpeng Zhu is a member of Dr. Qiang Zhao's team. He mainly is responsible for clinical research of Dr. Qiang Zhao's team. He is also in the cardiovascular surgery department of Region Hospital. So, as Dr. Jeff Drazen, made in the brief opening remarks, to actually paraphrase President John F. Kennedy, tonight, ask not what clinical trials can do for AI, ask what AI can do for clinical trials. So with that, I'm going to welcome the first speaker, Dr. Adam Dunn. So let me actually share my screen again and officially introduce Dr. Dunn. So Dr. Dunn's research is focused on applications of data science and machine learning in health. He has been an investigator or co-investigator on projects funded by National Health and the Medical Research Council of Australia and many other agencies. And he has published research articles and reviews in many top journals. And today, Dr. Adam Dunn is going to talk about using AI to improve the design, reporting, and the synthesis of clinical trials. So let's welcome Dr. Dunn.
Thank you very much, Zhenfei. So Zhenfei, would you like to show the video or would you like me to present instead?
So we are going to share the screen.
Very good. Thank you for the invitation to present today. My name is Adam Dunn and I lead Biomedical Informatics and Digital Health at the University of Sydney. Today I'm going to focus on applications of AI in clinical epidemiology and especially on natural language processing, NLP, which is my area of focus. Here in Australia, when we introduce ourselves, we like to acknowledge that the land we work and learn on belongs to its traditional owners. The University of Sydney campus sits on the land of the Gadigal, and I'd like to give my respects to the Gadigal elders past and present. I'd also like to quickly note that I do not have any financial conflicts of interest, but some of the research I'm going to talk about today was funded by the NHMRC in Australia, the NIH in the United States, and that I have other affiliations with societies and roles with journals and conferences that are relevant to the application of AI in clinical epidemiology. The way I'm going to approach the next 19 minutes is to split it into two sections. First, I will talk about how AI can be improved. It can be used to improve how clinical trials are designed and run individually. And then I'll talk about how we use NLP to improve how we synthesize trial results across many trials at once. Much of the theme of the talk will be about the gaps and connections between data and participants from trials and data and participants represented in electronic health records, or EHRs. I'll also be deliberate in going back in time to see how far we've come and to explain that we still have a long way to go to catch up to some of the ideas that were proposed in literature 10 to 20 years ago. At the top here, we have a quotation from a paper published in New England Journal of Medicine in 2011, discussing the value of patient data collected within hospitals and the process for informing the decisions that doctors make. This is a reminder for me to tell you my first summary point, which is that the gap between randomized controlled trials, RCTs, and retrospective cohort analyses from EHRs are going to get increasingly blurry, and that machine learning will play a key role in how we learn to replace parts of individual trials with routinely collected data from many patients. The image at the bottom here is from a paper by Raj Kumar and a team from Google, published in 2018, where text data from raw electronic health records in FHIR format were used to train models to predict mortality and diagnoses using machine learning. This is not a paper about clinical trials, but I like this image because it reminds us that EHRs can contain more information about a patient than would be recorded during a trial, that the data aren't perfect, and that much of the information is buried within clinical text and needs to be extracted and harmonized across locations. So here are some past examples of methods and applications that were designed to make clinical trials individually more efficient. In 1999, a paper presented at the AMIA Symposium that also had Zach Kohane as an author demonstrated a real-time method for paging trialists and clinicians using a rule-based system for patient eligibility. These are handcrafted rules for a specific trial. And so they might not be what we think of as AI, but it was a sign of things to come. This is also a great example because it made recruitment much more effective for the location where it was deployed. And it was based on the idea that it could also reduce bias in patient selection. In 2005, this stream of research became a clinical trial alert system built into an EHR system. In this case, it was an EPIC system, and it used clinical decision support system functionality and hand-coded rules for eligibility to create an alert that went straight to a clinician's inbox. Fast forward to 2016, and we now have at scale surveillance of over 1 million patients for phenotype and genotype information. This is still rule-based, but using much more sophisticated regular expressions in parts, and this system starts to work at scale to find eligible patients for precision oncology studies. In 2018, another presentation at an AMIA conference showed that if we only use structured information in EHRs, much of the important eligibility criteria for trials are not recorded. This wasn't the first to use NLP methods to extract data from clinicaltrials.gov, but it was interesting because it mapped concepts extracted from eligibility criteria from clinicaltrials.gov to concepts extracted directly from EHRs. In 2019, there was even a shared task as part of a national NLP clinical challenge looking at extracting information from clinical text to find cohorts of patients who could be included in a trial. So by this stage, NLP was firmly established as an important component for this application domain. From 2015, another closely related stream of research, one I was involved in, looked at ideas and methods for finding virtual cohorts from EHRs. In this case, not just for assessing clinical trial eligibility, but for replacing part or all of a clinical trial with evidence that was extracted directly from EHRs themselves. And in fact, if we are attempting to replace an entire trial, this is the idea of the green button that makes use of retrospective data rather than prospectively assigning patients to clinical trials. So in terms of how we use AI and especially NLP, these are the current trends I see in the area right now for how we improve the conduct and the design of individual trials. There's still a relatively consistent stream of research focused on matching patients to trials. And machine learning is used to represent EHRs and map them to trial eligibility criteria. There's also a certain number of companies or organizations that have been developed to build these methods and to sell them to clinical trial investigators to make these decisions more easy, more simple for them. I see less published research aimed at augmenting trial data with information from EHRs or from clinical notes in EHRs to reduce the burden of measurement during a clinical trial or to conduct long-term follow-up after trials are completed. This doesn't mean that the work isn't being done, it just means that it's less often being published in places that I'm seeing. For me, however, the most important application area now is related to replacing control or standard of care arms in studies with data extracted directly from EHRs. This is obviously an area of intense focus now because replacing control arms would substantially reduce the cost and impact of trials and make trials possible where recruitment was historically a critical barrier to completing. In terms of methodological research on NLP, the relevant fundamental tasks where advances may be of value include information extraction, document classification, and inference over multimodal data, and joint architectures over multimodal data for accurately filling in missing data in EHRs that would be important for recording as structured data and harmonized data as part of a trial. At the end of my presentation today, I'll come back to this list and tell you which of these I think are most worthy of your research attention right now. But first, let's go through how we use machine learning and NLP when combining results data across lots of trials at once. Around a decade ago, I was having a debate with Guy Safnat, Enrico Cuera, and Paul Glazio about how we should be automating systematic reviews. We landed on this idea of constructing systematic reviews as machines like player pianos that could watch how a human performed the individual steps of an individual systematic review and automatically repeat each of those steps over and over and over again at the press of a button. But these ideas were more practical than they were novel in 2013. In 2012, I proposed this new world of clinical trial data in a commentary piece in Science Translational Medicine. The idea would be that the identified and harmonized individual participant data from trials would be made available immediately, reported in computable forms as part of an ecosystem of meta-analysis tools, and quickly mapped to the systematic reviews and meta-analyses that are relevant. But it wasn't new in 2012 either. Ida Sim described and developed a global trial bank back in 2005, and she'd conceived of this even back as far as 1995. But back then, we weren't really ready to make clinical trial data available in ways that AI approaches could handle. The challenge is that if we focus on individual trials and individual systematic reviews, we end up exacerbating well-known biases, including publication bias, outcome reporting bias, and timing biases that mean that certain trial results appear more frequently in systematic reviews. And we end up basing policy and clinical decision-making on subsets of results from non-representative populations. There's also been a substantial amount of work looking at how we improve how to find trials to include in systematic reviews. This work stretches back to at least 2006, but in 2010 we had a bumper year. Boudin and colleagues wrote a well-cited article testing machine learning methods for extracting PICO elements from medical abstracts. Kirichenko tested methods for extracting key trial characteristics from full-text articles. And Wallace developed an active learning approach for finding trials that should be included in systematic reviews, where experts in systematic reviews would be fed with the specific articles that they needed to label and provide additional information for as part of the process. Fast forward to 2021, and Shifeng Liu from my team published a PICO extraction method at EMNLP that makes use of BERT, a pre-trained language representation model, and PICO extraction is still a very active area of research. In fact, it contributed as one of the problems that was used in one of the main well-cited examples of a BERT variant. In a related stream of research, Chunhua Weng's team were using clinicaltrials.gov data in 2014 to cluster similar trials, looking at patterns of trial eligibility. but also in 2020 created new shared data sets of trial eligibility information. And in 2021, the team had an article examining the differences between the EHRs of participants in OMOP format that matched eligibility criteria that were extracted from clinicaltrials.gov trials, or at least an extended version with additional information on top of data extracted from clinicaltrials.gov trials. So my group, together with Florence Bourgeois from the Computational Health Informatics program in Boston, have been working on methods for assigning trials to systematic reviews and safety meta-analyses by coupling information from trial registrations in clinicaltrials.gov and trial articles in PubMed, as well as connections between those and to systematic reviews in which they are included. In this main task, the aim is to rank trials from their registrations or published articles against a systematic review. That is, either a protocol for a systematic review, which is a bit like a cold start. We take a systematic review protocol and we try and identify which trials belong to that systematic review. Or if we have an existing systematic review that's been published and it has already included trials in it, we're looking for new trials that should be added to a systematic review if it was updated. And in that case, those trials are there ready to go as a kind of individual training. For systematic reviews of the efficacy and safety of treatments, they should be well specified in terms of their PICO, a description of the condition, the intervention and comparator, and the outcomes that are evaluated. So it's tempting to use modern language models like variants of BERT to extract neat versions of the PICO representation from the systematic review and from the trial and line them up. So we line up the PICO representation from the systematic review with the PICO representation extracted from the trial registration or article. And then we can take those PICO extractions and try and fit them together. However, we found that the more precise we try to be about the representation of the PICO, the worse the results. Across a big range of experiments, we found that very simple old methods where we take all the terms in those documents and use something like term frequency inverse document frequency to represent the terms as weights, that we get the best results. Just using basic machine learning methods across several distance measures between those aggregated weights. But none of those methods are good enough yet to completely remove the need for experts to decide on inclusion in systematic reviews. And none can beat active learning approaches, I discussed earlier from work by Wallace, that trains new models for every new systematic review with the help of experts. This means that currently the best tools we can hope for in this space are likely to be ones that monitor for new registrations and trial results and automatically send emails to systematic review authors to review maybe a handful of trials every week or so, and then add extra annotation and other information extraction tools to help them, systematic reviewers, quickly update meta-analyses and update their living systematic reviews so that the evidence is available as soon as the trial results are available to the public. So that was just one tool that we think can help make evidence synthesis faster, less wasteful, and to reduce bias. We developed ES3 as a kind of a sandbox for deploying all of these methods as tools in ways that can support crowdsourcing and sharing of data and manual validation of our efforts. Besides recommending trial registrations and articles to new review protocols like our cold start method, we also monitor for completed and ongoing trials above me in green and light blue in this picture that are recommended by our bots for adding to published systematic reviews. These can also be used as a kind of a nice indicator of when we think a systematic review might be out of date. We show similar and overlapping systematic reviews to systematic reviewers so that we can avoid doing redundant reviews where they're not needed. And we're also building intervention or treatment-specific pages that look for all relevant trial registrations and articles and systematic reviews and produce a cumulative meta-analysis about the safety of those treatments by extracting the structured results from the results data where they're available in clinicaltrials.gov. They might not be available for 100% of the available trials, but for those where we have data, we're able to make a summary of the safety meta-analysis from extracting information from the specific study arms. So in terms of synthesizing results across many trials at once, these are the trends that I'm seeing right now. The first is a range of papers that extract data from clinicaltrials.gov in bulk and use machine learning and often NLP to model some outcome, whether that's trial completion, additional later phase trials, publications, and so on. Some of these methods are quite useful, but many of them include flawed assumptions about the quality of the data that they're modeling. So I would urge caution here, because there seems to be a disconnect between the computer science advances where method novelty is much more important than the data quality and how that method might be deployed, and applied research in clinical epidemiology where the opposite is true. I've described the area about prospectively assigning trials to meta-analyses as soon as they're registered already, but the big one I think is about how we make the individual participant data, IPD, available for use as synthetic control or comparator arms in future trials, or potentially. If we can simply turn our ideas of what an RCT is upside down and run lots of single-arm studies against the aggregate of IPD from all prior trials. Relevant areas in NLP methods here include some similar tasks, including document classification, learning to rank, and perhaps pico extraction still has a role to play in data harmonization when we're trying to pool IPD across many trials at once. So quickly, here are my recommendations for where I think we should be focusing our efforts in NLP methods and applications right now. ClinicalTrials.gov is a treasure trove of important information. It's important that we can link it to PubMed, to Crossref, to all sorts of different areas, to results data and everything. But data entry in clinicaltrials.gov is nuanced, and measures of trial success are not as simple as extracting a categorical variable or a link to PubMed. Instead, we should continue to try and map out everything that's known about a treatment or an intervention or a condition and build tools to make results data available to everyone all at once. Trial and patient data everywhere are messy. And even with some impressive advances in NLP in the last 5 years, we're still a long way from being able to embed the contextual knowledge that we need to completely replace humans in decisions about patient eligibility, about construction of virtual cohorts, about whether a trial should belong in or should be included in a systematic review, and so on. Expert-in-the-loop solutions and simple tools that involve crowdsourcing are going to be needed for a while yet. So ignoring the importance of randomization for a second, the line between data for clinical trials and EHRs are going to become increasingly blurry. And beyond trials that are fully embedded in EHRs because of smart tools, machine learning is going to play an increasingly important role in finding patients and filling in information that's not recorded in EHRs. Finally, if I were an NLP expert and I had lots of money and time, I'd definitely be spending it on making as much de-identified and harmonized trial IPD available as I possibly could, because I think this is what has the potential to save billions of dollars and to transform clinical trials as we know them. So thank you for your time. This is part of the team of around 50 academics and postgrad that I lead at the University of Sydney, and we're looking for more. So if you're an expert in applications of machine learning and patient data and clinical informatics, I'd love to hear from you. And thank you very much.
Well, thank you, Adam. And we'll just have a little time for you and I to have a brief discussion before we move on to the next talk. So I have two major areas I'd like to get your input. You spoke a lot about analyzing trial results in the meta-analysis sphere, but as a dirty-handed experimentalist, I studied applied physics in undergraduate, and I was an experimentalist and liked to get things done. How useful has AI actually been in the recruitment of patients into clinical trials? As a clinical trialist, I can tell you that the thing that limits any clinical trial success is patient recruitment. Just like in real estate, it's location. In clinical trials, it's recruitment. So can you give us examples where it's actually worked? And if it makes a huge difference, why haven't people been adopting it to get clinical trials done quicker and with the right patients in them and get the patients to stay in the trials? You have to find them, convince them to join, and then keep them in the trial, especially a long-running trial. It may go on for years, as we see in many chronic diseases or cancer.
Jeff, I wish I had a better answer for you. I think a lot of the work that's being done in trying to improve recruitment and make recruitment more efficient is done outside of the world of publishing and is done by companies who are probably very good at embedding these kinds of technologies, and sometimes they're AI and sometimes they're not, into practice. But I don't think that the methods that they use are necessarily publishable, and I don't know that those things are things that I would see. So in terms of recruitment, I don't have a very good answer for you. But in terms of long-term follow-up of patients and in terms of trying to reduce the impact of a clinical trial on a participant, I think that's where the link with EHRs becomes important. And I think that's probably where machine learning is going to have a more important role to play, where we're trying to extract information from, say, clinical notes. I can't tell you an example where I think it's worked especially well. But what I can say is that the most recent publications that have looked at replacing control arms or standard of care arms in trials, which would be another way of reducing or improving the efficiency of clinical trials, they've been publishing case studies. And the case studies are showing that a full trial that includes a control arm and then simulates that control arm separately, they are producing similar results and they're getting representative populations. So there are case studies out there to show that we can make use of EHRs. And I think that machine learning and AI plays an important role in extracting information from EHRs.
Jeffrey, do we have time for one more question? I show a lot of shaking your head, yes. So one of the things that is really lacking in the world of clinical trials are large numbers. We publish a clinical trial, and I think that the best example was currently that everybody in the world knows about, the COVID vaccines. The COVID vaccine trials had 30,000, 40,000 people in them, and then they start giving the vaccines to millions, and suddenly things start popping up. People develop myocarditis. There's sort of a weird clotting stuff that goes on. So to what extent has AI been useful in the follow-on of clinical trials? We have a medicine that we think works, but there can be rare events that can be devastating, but we don't have any idea of their frequency. So has AI been used in the follow-on here to help identify those adverse events that might not have been cropped up, and how successful has that been?
I don't know of examples where AI has been used to improve the follow-up following clinical trials, but we certainly are looking at applications of machine learning in trying to improve how we look at safety of approved drugs and treatments. Look, it's such a long and complicated area. Florence and I have been working on trying to look at this for quite a while by extracting information from trials and clinicaltrials.gov as a kind of adjunct to or replacement for adverse event reporting systems and Sentinel. And I know when I visited the Landmark Center, the Sentinel program was downstairs from where I was sitting in the Landmark Center in Boston. I haven't seen examples where we've done these, what's the right word for the mega trials? I'm sure there was a paper that was published a few years ago where they were talking about extremely large scale trials that were fully embedded in electronic health records. And I don't know that any of those very, very large trials make use of machine learning and AI. They just make use of big data. So in terms of, if I was to answer your question very directly, I'd say I have no idea, but I expect it'll be on its way.
Okay, Jean-Fei, I'll turn it over to you to introduce our second speaker.
Sure. So our next speaker is Professor Zhao, and Professor Zhao's clinical work focuses on the adult cardiac surgery in the fields of coronary artery heart valve disease, aortic disease, and heart failure. He developed many modern cardiac surgery techniques in China. And as co-PI or leading PI, he participated in several clinical trials such as coronary, CAMPAS, and so on. And so Dr. Yunpeng Zhu is a member of Dr. Zhao's team, as I already mentioned. So let's welcome Dr. Zhao, and so we're going to play the video.
Dear Dr. Drazen, Dr. Rubin, Dr. Hamel, distinguished editors, professors, and colleagues, it's my great honor to introduce our AI-assisted QFR-guided trial, quantitative flow ratio guided revascularization strategy for patients undergoing pulmonary valve surgery with comorbidity coronary artery disease, Phase 4 QVAS trial. Valvular heart disease, VHD, is common in the world. In the United States, the prevalence of undiagnosed moderate to severe valve disease was 6.4%, as 2021 AHA reported. More than 78,000 surgical and 85,000 transcatheter valve procedures were undertaken in 2019 from STS database. In China, the weighted prevalence of valve disease was 3.8%, with an estimated 25 million patients in 2015, and more than 66,000 surgical and 6,500 transcatheter valve procedures performed in China in 2020. The valve heart disease guideline recommended coronary angiography screen before valve intervention for patients with symptoms of angina, objective evidence of ischemia, decreased left ventricular systolic function, history of coronary artery disease, man over 40 years old, and a postmenopausal woman. It was reported that approximately 20% patients undergoing valve surgery have comorbidity coronary artery disease, stenosis over 50%, especially in elderly population with degenerative valve disease. The guidelines also recommended combined CABG in the valve surgery. If left main stenosis over 50% or major coronary arteries over 70% as class 2 recommendation, according to 2020 ACC-AHA valve guideline. And if major coronary artery stenosis over 70% as a class 1 recommendation, while 50 to 70% stenosis as class 2 recommendation, according to 2020 ESC-EACTS valve guidelines. The all evidence is level C. Let me present a case. This is a 70-year-old gentleman with degenerative severe mitral regurgitation, NYHA class 3, admitted for mitral valve repair. Preoperative coronary angiogram found left circumflex stenosis of 60%, without angina. Following the current guideline, combined CABG with mitral valve repair is indicated to achieve complete revascularization. However, it will make the procedure more complex, increasing surgical trauma, cardiopulmonary bypass, and aortic cross-clamping time, which means more cardiac ischemia. On the other hand, if the territory has no ischemia as a fact, the combined CABG will increase the risk of graft failure due to competitive flow and also accelerate the occlusion of the native coronary artery. This is real-world data. The STS database showed the mortality and the major morbidity for combined valve procedure plus CABG is much higher, 2 to 4 times than isolated valve surgery. In STS database, operative mortality is defined as death within 30 days after surgery. Major morbidity includes stroke, renal failure, which is defined as new dialysis, and so on. For example, the operative mortality of aortic valve replacement alone was 1.9%, while aortic valve replacement plus CABG was 3.6%, almost twice. Mitral valve replacement alone was 4.5%, while mitral valve replacement plus CABG was 9.6%, more than 2 times. Mitral valve repair alone was the lowest, 1.2%, while mitral valve repair plus CABG was 5.4%, more than 4 times. The guideline of coronary revascularization strategy is shifting from anatomical to physiological. The fractional flow resolve FFR is the most commonly used physiological methods of assessing lesion significance. The role of FFR in guiding PCI for stable patients is solid, with significant clinical improvement. However, the role of FFR in guiding isolated CABG is uncertain. Benefits were observed from retrospective studies, but wasn't confirmed by small randomized clinical trials. Quantitative flow ratio QFR is a novel approach to compute FFR from angiograph imaging. No need for pressure valve and adenosine. QFR has been extensively validated and showed good diagnostic concordance with wire-based FFR, with a sensitivity 85% and specificity 91% of the first generation technology. The recently published Figure 3 China study showed that QFR-guided PCI improved 1-year clinical outcomes compared with the standard angiographic guidance, with about 1/3 reduction of MACE at 1-year follow-up. The new generation of QFR, named as Mu QFR, is powered by AI and allows computation of QFR from a single angiographic view. With a high automation achieved by AI, the computation time is dramatically reduced to only 1 minute. All side branches, where the most grafts are put on, are simultaneously analyzed, which is really useful to guide CABG. There was also excellent diagnostic accuracy of AI QFR compared to wire-based FFR. So, the rationale of QVAS trial is that patients undergoing valve surgery with combined stable coronary artery disease may benefit from physiologically guided revascularization decision-making. We believe that AI QFR will be the first choice in this surgical situation. The advantages of this new technology are non-invasive, no need for pressure wire and adenosine, no additional trauma. Convenient, no need cardiologists do more than conventional coronary angiogram. Efficient, QFR calculation can be done within 1 minute. Integrative, simultaneous assessment of multiple branches. This is the overall looking of our trial. There were 4 QVAS, QFR-guided CABG in valve surgery. This is a standard prospective, multicenter, randomized, blind, parallel controlled trial. To our knowledge, this is the first randomized clinical trial using AI QFR technology. Also, the first QFR-guided surgical revascularization trial worldwide. Major inclusion criteria: patients undergoing primary valve surgery with comorbidity coronary artery disease, at least one vessel stenosis over 50%. Major exclusion criteria: secondary valve disease, such as ischemic mitral regurgitation, planned transcatheter valve procedure, planned PCI, emergency or lethal cases. Patients will be randomized to 2 arms, 1-to-1. QFR-guided arm as an intervention. CABG is carried out for suitable target vessel with lesions only if QFR less than 0.8. CABG-guided arm as the control. CABG is carried out for every suitable target vessel with lesions stenosis over 50% by visual estimation on coronary angiogram. Our pilot study showed revascularization strategies were modified in about half cases and free of combined CABG in 1/4 cases following QFR guidelines. The primary outcome is defined as MACE-5, a composite of all-cause deaths, myocardial infarction, stroke, unplanned revascularization, new dialysis within 30 days after surgery. The setting of this primary outcome was based on STS database, as the incidence of these individual endpoints could easily be found in the database as reference. The secondary outcomes include MACE within 1 year and at 3 years, graft failure at 1 year and 3 years, EQ5D score at 30 days, 1 year and 3 years, cost effectiveness at 30 days, 1 year and 3 years. Randomization is done by IWRA system and is stratified by site with fixed block. Blinding is triple for patients, surgeons, and endpoint evaluators. Surgeons are informed graft targets the day of the procedure, but not how the decision was made and in which group the patient is The bypass plan is determined and the patient is randomized by the unblind core lab. Statistical hypothesis was based on STS database. The expected incidence of primary outcome is 12% in CABG-guided arm and 6% in QFR-guided arm. The risk ratio is 0.5. Of course, this is a superior test. Totally, 792 patients will be enrolled. This is an investigator-initiated trial sponsored by Beijing Hospital, Shanghai Jiao Tong University School of Medicine. The core lab is Pulse Medical Joint Laboratory, Shanghai Jiao Tong University. The data manager is Biostatistics Center of Fudan University. CRO is DMED. SMO is AES of PPD company. This trial is funded by Beijing Hospital, Jiao Tong University School of Medicine, and the Chinese government. The study timeline. The trial was approved by IRB in 2019. The pilot phase enrollment was completed in our hospital from 2019 to 2020. The multi-center enrollment is from 2020 to 2023. So far, 331 patients have been enrolled from 7 sites. The actual incidence of primary outcome is 10.8%, close to the hypothesis 9%. The primary outcomes of surgeries will be expected to report in 2023 to 2024, and the secondary outcome of 3 years will be reported in 2026 to 2027. Okay, let's back to the case I previously mentioned. After AI QFR analysis, we found that there was a diffuse lesion in the left circumflex. The maximal diameter stenosis was 45%, and the maximal area stenosis was 70%. The QFR value of the whole left circumflex was 0.82. So, if this case, following the CABG-guided strategy and the current guidelines, the surgical plan will be mitral valve repair plus CABG. And if following the QFR-guided strategy, the surgical plan will be mitral valve alone. Summary: valve heart disease with coronary artery disease is common in elderly patients. The evidence gap of combined CABG exists. AI QFR is a useful tool for accurate assessment of coronary physiology. Favor 4 QVAS trial may reduce MACE of valvular heart surgery with CABG. Randomized clinical trials with large population and long-term follow-up are needed. Thank you for your attention.
Thank you, Dr. Zhao. And I will ask a couple of questions, and if other people have questions, please feel free to raise your hand using the Zoom tool, and I'll try to call on you. So my first question is, this is a fascinating and important trial. You're actually testing AI using standard trial methodology, and I congratulate you for doing that. It's important to test to see if what we think is going to be an advance really is. there is a German word, Schlimmbesserung. It means an improvement that makes things worse. And so we have to be very careful whenever we institute something to make sure that it's an improvement that actually makes things better. So in your trial, which is ongoing, and I congratulate you for getting it started during COVID in a place and time where there's been a lot of lockdown off and on, and it must have been very difficult and something that you hadn't planned for. But two kinds of questions come up. One is, are you using AI at all in your recruitment to find these patients, as suggested by Adam in his talk? Or are you using standard methods to recruit patients for your trial?
Dr. Drazen, thank you for the question. Actually, the quantitative flow ratio is analyzed by AI, not patient enrollment.
So that everybody who is having an angiogram gets the AI, and that has a lesion, then gets the AI analysis. Is that correct?
Yes.
So, do you know what fraction of people who would qualify for the trial by their medicine agree to join?
In our center, is about 50% to willing to be in the trial.
And is it the doc? Sometimes we find in these surgical trials, it's the patient's physician who's convinced, for example, that the AI-guided flow reserve is going to be better than the human calculation, and they don't want their patient in the trial because they want them to get the better thing. And there are other people who feel the other way around. So can you tell me whether it's the patient or their physicians that are giving you the no answer and half the patients that aren't agreeing to be in the trial?
I would say the surgeons will ask a patient to be involved.
Mm-hmm. So the surgeons are fully in favor of this, as far as you can tell.
Yes, because there's no certain evidence the additional CABG is good or bad.
Mm-hmm. Now, when you went over your original stats, you showed that in the U.S., about half of aortic valve surgery is now done using the transcatheter approach. Actually, a little more than half, and the other half is being done by standard open methods. And I imagine that the two types of patients in the two groups were probably not physiologically matched. My guess is that the younger patients are the ones getting the standard open heart surgery, and the older, more comorbid patients are the ones receiving the transcatheter valve surgery. While in China, at least from the data you showed, transcatheter valve surgery has really not reached the levels that it has in the U.S. data. So how do you factor in whether a patient in your trial may be better off with a totally different kind of valvular intervention rather than a surgical valve intervention, a transcatheter valve intervention, in which case they could have stents placed. The entire thing could be done in the cath lab as opposed to in the operating room. So how do you make that decision for each individual patient, and how does AI play a role in
that? The patient is suitable for TAVI transcatheter intervention is excluded in this trial.
And who makes that decision?
The physician.
Right. So if a physician thinks that a person is better for transcatheter valve, then they're not even allowed to participate in the trial.
Yes.
They have to be someone. But right now, transcatheter valves are not all that common in China, whether it's you don't have the expertise or you have much better surgical technique or much more of surgical availability. We'll see how this trial plays out. But that, of course, will be what I call an ecological change. As you designed the trial 3 years ago, as far as I can tell, during the period between 3 years and the next 3 years when I think you're going to finish your last intervention, looks to me like your last intervention will be sometime in 2023. Is that the way you think it will play out, or do you think it'll speed up now that there's less problems with COVID?
The enrollment is slow right now, but the TAVI number is still behind the surgical patients because of financial issue in China. For younger patients, I would say the SAVA is better, much better. The clinical, the long-term results is much better than TAVI in young patients. So,
and of course, the younger patients are going to have less coronary disease. So it'll be interesting to see how it plays out. It's a very interesting trial, and I congratulate you on working through this problem to test it in a way that makes sense, at least at the time it was designed. And hopefully the information that we gathered from it will be useful. I'm impressed that the Q-related, the mu QFR, the data can be available in real time, as we say, to help you in decision making in the cath lab or in the OR. So with that, and having not seen other questions arise or hands risen, I'm going to turn it back to Jean-Fei to do our introduction of Dr. Chatterjee.
Sure. So our third speaker is Dr. Arnaub Chatterjee. And Dr. Chatterjee was an advisor to Chief Technology Officer at the U.S. Department of Health and Human Services during the Obama administration, as already introduced. And his work has been published in journals such as the Harvard Business Review and Nature. And he also had held board appointments at Cornell University and the Global Future Council of Biotechnology for the World Economic Forum. And today, so he is going to talk about applications of AI to improve trial design and the probability of success. He's actually in Colorado right now, so it's very early for him. So, Dr. Chatterjee?
Yeah, thank you very much. And I'll say this with humility, given the audience I have. I'm not a physician, so let me take that back as a credential. But I do spend a lot of time working with physicians and physician scientists. And what I'll talk about today is actually the perspective of one of the companies that are operating in this space as an AI company. There we go. So, and I'll talk about I'll set up the talk by presenting, what I think are different trends that I've seen over the last 12 years in my roles. I've spent time working at McKinsey, at Merck, with the launch of Keytruda in immuno-oncology, and then currently at Medidata, where I work with about 40 or 50 different biotechnology companies or pharmaceutical companies in trial design, and maybe set up a set of beliefs that are a little bit pointed, but hope to get to kind of how I see AI applications taking place. And then the last part of my talk will be focused on actual applications with actual companies in a range of different AI applications, starting with what I'm calling not AI at all, but kind of established biostatistics methods, moving more into predictive modeling and deep learning. All of this in the spirit of how do you improve the probability of success within clinical trials and de-risk the actual process. So just to tee up, what I do and kind of the way we think about data, which is important. Metadata, where I work, is a 23-year-old company that's the underlying software to capture data through the case report forms for clinical trials. So we digitize the information that an investigator or a PI or a site might put into a clinical trial form. And we currently do this for over 50% of the world's clinical trials. So our customers include pharma, biotech, large academic medical centers, and med device companies. So we serve actually most of the world's organizations who are running clinical trials. And as part of our relationship with these organizations, the vast majority of them have agreed to share both operational and patient-level clinical trial data with us. So that's the basis for what you're seeing here, Acorn. This is a roughly 300-person organization that I lead within Metadata. This is the data science and AI company that reuses historical clinical trial data in an aggregated and anonymized way. And we're helping companies make more informed decisions in drug development using different types of data science methodologies. So what you see on the left-hand side is a large patient population of 8 million patients. It is all historical clinical trial data. It's coming directly from the case report forms in a completely raw way. And this is the data we use as the basis for how do we think about the trial itself. So can we use historical data to improve protocols, predict patient outcomes, or create synthetic control arms or synthetic patients, which Adam previously had referenced. And I'll give some examples there. And to Jeff's previous point around how do we think about AI in operations or in trial enrollment, that's another area that we focus on as well. So can you identify patients earlier? Can you improve site recruitment using different types of methodologies? And then the area on the right is the areas that we play in, which is trial operations, protocol, and then how do we get drugs to market more successfully. So I want to start maybe with a set of bold opinions here, but I wanted to share where I'm coming from and kind of how I arrived at these statements. I think, drug development has always been inherently risky. There's a multitude of risks that are both operational and clinical in nature that hamper and challenge the likelihood of approval or a regulatory milestone. And the key is, once you have a biological validation, which is ideally a molecule that hits against a protein target that's part of a disease, it goes through a series of different hurdles, which is typically expensive and time inefficient. I also think, in the biotech world right now, we're seeing a real lack of clinical trial success in recent months and relatively this past year, despite signs of early biological promise by a lot of companies. And I think why I'm arriving at these conclusions over the last 12 years of work is that, I found the process of making protocol decisions or trial design decisions happens in a siloed and rather data uninformed manner. So when I use the word hubris on my slide here, I found that, the program leads within biotech companies or pharmaceutical companies, chief medical officers, they all grapple with the same set of questions, but definitively place their bets based on prior experience, published literature. And I think what's exciting is that the data science world has evolved considerably in a relatively short period of time. Machine learning has evolved, and it's become easier and easier to pick the winners. You can analyze by company which initiatives are having a real impact on R&D and who is using data in a very progressive way, which is what I'll talk about. Go back a slide. So these are two charts that are derived from work that Andrew Lowe of MIT has done. And he's pulled basically kind of the most recent picture of in-phase success of clinical trials based on data from 2011 to 2020. And you can see across different therapeutic areas and different indications, still very low levels of approval in the earliest phases of trials when demonstrating safety or optimizing the dosing are critical. And then overall likelihood of approval, especially in oncology and cardiovascular trials, continues to be low. The other challenge here is you're seeing a shift in the market towards more complex biological pathways, which have greater therapeutic potential, but there's also greater chances of failure. And that's caused drug development to be increasingly risky in the past decade. So the key stumbling blocks here are really around demonstrating efficacy. Companies are including flawed study designs or inappropriate statistical endpoints. They might be underpowering their clinical trial, small sample sizes. So this is what's resulting in patient dropouts, insufficient enrollment. And then the other barrier is obviously safety, where it's critical in every phase of a trial, but safety is only really apparent in some of the larger populations that are associated with phase 3 or post-approval studies, such as phase 4. And I think what's important here is, at the highest level, drug developers are making a set of decisions that are tied to a few different things. So one of them is around clinical trial design. What are the key primary and secondary endpoints, the number of arms, the inclusion-exclusion criteria, what comparator do you use, and use of biomarker, and so on. And then you can kind of see the regulatory piece is also incredibly important here, where prior approvals in indications or therapeutic areas are certainly a path forward. Breakthrough therapy designations, accelerated approvals as determined by the Food and Drug Administration do create a way for companies to shift their mindsets around how they make trial design decisions. And then all important now is the drug biology, the mechanism of action, genetic or any epigenetic alterations, target gene expressions. These all tie into a set of different decisions that companies have to make. And these are kind of the key buckets of the types of risk trade-offs that you have to make. And this gets increasingly complicated. It's not a straightforward process. So this is kind of an expansion of the previous slide, where you can kind of see the multitude of different scenarios and decisions that are critical to moving an asset forward in drug development. And the clinical sub-team within companies are responsible in an evolving and multi-scenario way to design a trial, understand patient stratification, and seek cross-functional input from a bunch of different stakeholders within their companies. And this is all reflective of a changing dynamic in a scientific field. If you have a new mechanism of action emerging, or if you have a new product approval, or it's a change in the operational and the commercial landscape. In competitive therapeutic areas, it's increasingly difficult to recruit patients, so that changes how you might make a decision. Or in therapeutic areas that might already have entrenched or well-established, first or second line products, that also changes your decision. So the idea here is, how do you align all of these decisions along cost and time and quality dimensions? And this is where data science can play a real role, and it can move the needle in a very robust way. I wanted to kind of play in a real life example here for you, for a company I worked with recently. This is in a really, really crowded space in inflammation. And this is an anecdotal example, but one that I thought really kind of exemplified how difficult this decision is. So within this company is focusing on a phase 3 launch, and there are 50 different drugs and 8 different classes of drugs within the inflammation space. There are currently 10 head-to-head trials, and there are very established incumbents in this space, not only clinically, but also commercially. So you might have a drug like Skyrizi, which is the branded name, but this is an efficacious drug by AbbVie that also has very good market access. There's Tremfya, which has a cost access. There's Cosentyx, which has shown a strong clinical profile. And what we know is that the drug that we were working with here had a strong target profile. It had superiority to certain products. It achieved skin clearance, within plaque psoriasis at a certain period of time, clean safety profile. But it's not straightforward in how they would think about placing themselves commercially, or how they might think about how to position their drug from a safety perspective. So the key approach that they're taking here, and the data that they wanted to move forward on, was, is there a particular unmet medical need that addressed, the lack of oral therapies or topical therapies in the market? There are specific patient populations of interest that are currently untargeted by the existing products. So you're really getting to a specific set of patient populations that you can target as a way to win. And you're looking at just a series of different trade-offs where you're trying to build a strong evidence profile for your product, but also realizing that is a pretty difficult set of trade-offs. I'd be remiss if I didn't talk about the existing data in the world to help inform these decisions. Like I mentioned, my company works majority of the time in historical trial data, where that you're seeing lower volumes, you're seeing much more standardized and robust collection methods, and it's complete. We're working directly with the case report forms that companies are sharing with us in confidence, and we have the entire patient longitudinal view for that trial. And as a result, we have received favorable opinions from regulatory agencies around how we might use this data and how it could be used for certain data science applications. However, we know that trial populations are not representative of real-world populations, and despite the growing amount of data in EHRs and genomics and claims data, it's still difficult to understand how to use this in a very robust way due to different types of bias that might exist. So we want our treatments designed in the trials to be representative of what's happening in real-world settings. It's not always the case in terms of what we see within the safety and the efficacy. So as the primer, I'm going to set this up as sort of 3 different areas where we can de-risk clinical trials using 3 different methodologies and 3 different companies that have done this work. And this is all work that we've done kind of with this historical clinical trial data that's been aggregated and then answering a series of questions that I was previously laying out to inform a regulatory decision and improve the probability of success. And I'll focus less on the data science methods and more on what are the use cases that people are trying to solve for and how is AI being used. This first example is actually not AI at all. It's intended to show the power of robust data as well as well-established biostatistical methods and how it all comes together to save time, to improve confidence in study design and move a regulatory needle. Adam referenced this concept of a synthetic control arm, or what's also called an external control arm. It's a relatively novel methodology. It's been growing in popularity over the last couple of years, but it's an idea that if you can statistically match from a vast pool of individual patient-level data coming from EHRs or coming from historical trial patients, you can tie it to the experimental arm of a current-day clinical trial. And a synthetic control arm can augment the number of patients in your control arm, or in some cases, it can replace it entirely. But if you do this right and you do it well, there's a lot around creating estimation and confidence in understanding and interpreting your trial, even kind of deciding which therapy you want to advance. And so the promise is very exciting. The methodology here is not complicated. It's a propensity score matching methodology and a weighting methodology that is a commonly accepted type of method accepted by the FDA or by the EMA right now. And if you have achieved really good baseline characteristics between the synthetic control and the randomized control from the target trial, you can replicate the control arm of the target trial. And this can help mitigate a lot of challenges that are faced with a randomized control in really difficult to study indications. So in this particular case, this is with Kite Pharma, which is a Gilead company. It was work that was presented at the American Society of Hematology last year. And they were aiming to use historical trial data to contextualize the results of their experimental therapy in ALL and compare overall survival and overall complete response rate at 24 weeks for their pivotal trial. So they used historical trial data that matched the inclusion and the exclusion trial criteria from their target trial. And they wanted to better understand how to create a synthetic control arm that was constructed with different criteria such as ECOG scores, Philadelphia chromosome status, bone marrow blasts, ECOG, or age and sex and other demographics. So 40 total patients were matched in this trial. And effectively what we did was show a clinically meaningful treatment effect that was across all endpoints that were attributable to the experimental therapy. And it shows strong evidence for its use in adult patients with relapsed or refractory ALL. And the comparison of overall survival was very strong. We saw a much higher median OS in the experimental therapy versus what we saw in the control arm. And this was all well validated. And this company will likely move forward in using this in a number of different regulatory settings. But what it showed is that it's possible this is a light AI model, if you will, of how you can use this data to move needles. The other thing I'd say is that, in areas like recurrent glioblastoma, in other oncology indications and in rare diseases, synthetic control arms are becoming very popular and regulatory accepted. We've seen 2 different acceptance and approvals for recurrent glioblastoma by the FDA now for use of a synthetic control in a phase 3 design. The second methodology and example is actually in the CAR T space. And I've been thinking about this a lot, but if you look at some of the failures in CAR T recently, they're due to a bunch of different reasons tied to safety, where development was stopped after the death of a handful of patients. We saw something like immune effector cell-associated neurotoxicity syndrome, or ICANS. in many trials for prostate cancer for one of the CAR T companies. We saw chromosomal abnormalities that were observed in patients during bone marrow biopsies. Safety continues to be the main question here. And despite the huge impact of CAR T adverse related events on clinical development and success, there's no approved or validated or widely used way to predict these types of events. So ideally, we would want to use better data science methodology and predictive modeling to understand the toxicity profile of a therapy. Can we do a better assessment of ICANS or cytopenia or cytokine release syndrome within patient subpopulations? And then can we truly differentiate a treatment-related adverse event versus other adverse events within certain patients? So this data challenge is a real one within CAR T. Work that we've done here actually was, can we use our historical trial data, which spans about 2,500 CAR T patients, 400 of whom we had severe cytokine release syndrome for, and create predictive models to understand what contributes to CRS. So this is an example where we were able to tie different data science methods such as XGBoost, which is a machine learning model, to better understand, can this predictive model take very important multi-domain features and predict with accuracy CRS risk across patients in that training data set. And the lab data ended up being incredibly important here. Some of the most important results that we found that were consistent with literature were around mean platelet levels that were lower among patients with severe CRS, which is what the literature has said. We also understandably found that patients with high disease burden were at increased risk of CRS, especially when their platelet counts were low. The other observation here is around really zeroing in on all the severe CRS patients that we found in a really, really narrow zone of low neutrophil and platelet scores, which were driven by the same hit to hematopoietic function. And this is consistent with literature, but not something that this company that we worked with understood very well. So the takeaway here, the key point is that predictive models were accurate in assessing, could we find patients who exhibited an adverse event that's commonly found in patient populations with CAR T? And can we learn what's the underlying basis for those adverse events and then decide how we might want to think about enrollment or how we might want to avoid that adverse event moving forward in terms of the screening criteria for those trials? Now, I'll take one last example here, and this is a little bit art of the possible. It's early day technology, but this is built on something emerging in deep learning around what's called synthetically built patients, or what you might have heard is called digital twins. And, the idea of trial simulation has been around for a long time. This is a whole different level of machine learning to try and understand, can you build an in silico individual representation of a patient that reflects their molecular or their physiological or their lifestyle status across time. And it's envisioned that, we can use this type of synthetic patient model to create virtual experiments. We can, kind of do it in an iterative way where we're trying to answer a bunch of different questions, predicting a trajectory for a patient's care compared to standard of care or different treatment variations. So we built a new synthetic patient technology that leverages deep learning. It creates a high fidelity synthetic data set that's, again, built from our clinical trial source data. It conceals identity. It is actually tested against a number of other synthetic patient technologies that are currently available, and a number of academic groups are working on this. But recurrent neural networks, GANs, these are other models that are attempting to do this, and we found our methods to be more accurate and with comparable or better performance. So this particular pharmaceutical company was launching a new TKI drug in non-small cell lung cancer. They wanted to use a synthetic patient data set to understand why some trials show higher effects than others for the same drug. They also wanted to support phase 3 trial decisions, such as understanding geography or comparators or subpopulations. And you can see from the Kaplan-Meier diagram on the right that a synthetic data set that we built that picked, some of the core baseline characteristics closely mirrored progression-free survival outcomes and overall survival for the comparator drug. And for this company, we're using this data set to answer a few different questions. How likely is their drug going to demonstrate comparative benefit in all of the primary outcome measures, such as overall survival or progression-free survival? Can they make a legitimate claim of superiority or non-inferiority compared to other drugs that are in the market or standard of care? And then can we understand, is there a way for us to better understand which patient populations would represent current standard of care and what can they do to target their patients differently? So can different methodologies in AI change the inclusion and the exclusion criteria faithfully that recapitulates the real world population that's going to derive any benefit from their drug? So this is, again, early days. It's we're publishing this, sharing it at ASCO actually in a couple of weeks, and I think it kind of sort of paints the art of the possible, if you will. So I'm going to stop there. Hopefully these were depictive examples, but the goal is to show companies doing things in this space that significantly can move the needle, save real time or cost, or improve the way that we think about recruitment. And then how do we start to better do this in a scalable and iterative way?
Well, so thank you, Arnaub. And it's interesting to see how this could move forward. And I just have a few questions that came to my mind as you were speaking. And first of all, with respect to controls, since I'm sort of naive in this area, are you aware of studies where people have predicted what the controls would be like and then compared them to actual controls? In a prospective fashion in the course of a trial, you would say that these were the controls that we randomized to control, and here's the control group that we simulated. How close are those two groups, both in the point estimates and in the variability around that estimate, which usually is the backbreaker in clinical trials?
Yeah, I think that's a really good point. So we have actually started to take our trial data to prospectively design control arms for companies that are designing a trial right now. So the idea is that you can take aggregated data, you can break down the baseline characteristics of these trials, mirror the target trial criteria pretty accurately, where you're getting almost perfect overlap, in the design of the target trial. And ideally, you can actually model and simulate different control arms based on different trial criteria that you're modeling for. So it's not a widely accepted methodology yet. I think most people come in with a predefined idea of who they believe the target criteria should be and what the control arm should be. But theoretically, if you take previous trials data, you can actually successfully do this. And this isn't AI methodology at this point. These are pretty rote kind of propensity score weighting methods that can derive those types of estimates.
And so then, the second question comes up is how robust and how easy is it to tune out and tune in what you're looking for? Because, there are two different kinds of goals here. And I hate to sound sort of jaded, but after 19 years at the New England Journal, I can tell you there's a reason to be jaded. Can you tune your control group so that it's most favorable to the actual group, or can what we learned about AI models is that small differences can end up at the beginning with large differences in the prediction. So you want your drug to look good, so the easiest way to make your drug look good is to make the control group look bad. So you just tune your model just a little bit, something that you wouldn't miss at all, and as you're tuning the model, you discover that if you include people who are 3 months older or have, some comorbidity, that they do much worse. And so you include those folks in your model design. So how do you avoid that temptation? And can you then kind of retune your model for the people who are actually enrolled? These people who are actually enrolled, and how can you do it? So how do you avoid that kind of bias?
Yeah, I think you have to. There's a few things here. You cannot. You have to blind yourself to the outcomes. You can't cherry pick. So when we've seen, for example, in single arm trials, there was recently a company in the newly diagnosed glioblastoma space that submitted a synthetic control arm. But where I think they got dinged by the Food and Drug Administration was because they looked at outcomes before having finished the single arm trial. This is a big problem. It'll cause the FDA to say that you cherry picked control arm patients. Right. And I think that's where you have to be blinded to the outcomes, prior to the actual submission. So what we're finding is that before these designs are submitted, you have to pre-specify the criteria. You have to show the design to the FDA or the EMA. They have to sign off on it. And this is an operational hurdle, but most companies don't do this. They actually submit these hybrid designs or these novel trial designs without any sort of pre-specification or FDA input. And they get, for the reasons you're describing, they get in trouble. And I think that's problematic. But you have to obey some code of conduct as it relates to not over-tuning. Let me put it that way.
So we're running late, so we need to move on. Doctor, you have some questions, and Dr. Zhou, when we do the roundtable discussion, you'll have your opportunity to ask these questions. I know that you have had a number of them. And so with that, I'm going to turn it back to Jean-Fei to reintroduce the panel members, and then that will give us about 30 minutes for a discussion. What way this is going to work is that Jean-Fei is going to introduce each person, and then each member of the panel will have 2 to 3 minutes to give her or his ideas of importance of what they've heard in this space and how it can move things forward. And after we've heard from each of the people individually, we'll have a discussion among the group. So with that, Jean-Fei, the floor is yours.
Sure. So I think the majority of people, the distinguished guests, have been introduced in details, but I would like to particularly introduce again Dr. Junbo Ge. So he is a member of the Chinese Academy of Sciences and professor and director of Department of Cardiology and Interventional Cardiology at Zhongshan Hospital in Shanghai. And so academician Ge has made great contributions to diagnosis of coronary disease by intravascular ultrasound. And as the primary investigator, he has won many awards, including the second prize of the National Science and Technology Advancement Award, and so on. So with that, I think as Jeff just said, I think Dr. Ge has several questions to ask, I think, during the presentations.
Thank you very much, Jeffrey. Actually, very nice to attend this meeting, Dr. Drazen. I still remember we were at your house for the dinner. I still remember all the friends, meet all the friends and your board members in your home, in your garden. Actually, my thought regarding this today's meeting. Actually, I thought it's what I imagine. What is giving the talk by Dr. Chatterjee regarding how to design using AI. Actually, before I ask the question, I would like to give some comments regarding the TAVI in China. 12 years ago, roughly 12 years ago, I introduced this technique to China. I did the first TAVI in China, and with the help of our cardiac surgeon. The problem is before 2017, we were not able to acquire the equipment. We have to really buy the equipment from Hong Kong and deliver this to mainland China, and so only limited hospitals could perform this procedure. And I have normally on the weekend travel here and there. They prepare the patient. I do the case there. And since 2017, we have just this time, 2017, just by the Labor Day, the 1st of May, within 1 week, the SFDA approved 2 products. One is the Venus A, the self-expanding valve, and the other is the J valve, the transapical. We have our device, and since then, we have many centers now doing this transcatheter valve replacement. Up to now, up to last week, we had the data. We have 18,000 cases performed in last 12 years, and many in last 5 or 6 years, because as I mentioned, since 2017. Therefore, the majority of the patients in China with valvular disease are still performed by our cardiac surgeon, like Professor Zhao, by the top surgeon. This is the first comment. Secondly, what the trial Dr. Zhao just presented regarding the QFR-guided CABG versus CABG plus valve repair. Actually, this is not, I thought, the AI design to improve the design of the clinical trial. Actually, it's the AI assisted to interpret ischemia, because this parameter, Dr. Drazen, maybe you know, in 1996, I believe, in New Jersey Medicine, Dr. Bernard Debreu and Nico Piles, they published the first paper regarding FFR-guided intervention to using FFR to interpret ischemia versus angiogram. They're using at that time the 0.75 as a cut point to do intervention or not. And now we in the era of DS, we're using 0.88. This parameter designed by developed by Professor Tu Shengxian, actually is QFR. This QFR is using the software to calculate the FFR or ratio or reserve to replace the guided calculated flow reserve. Therefore, I think this is a very smart design to using this a new QFR to interpret actually ischemia or the angio-guided the severity of stenosis. I think this will give the answer. I am not sure 2 or 3 years after, when the majority of the patients in China with valvular disease go to the intervention, not for example, surgical repairment or replacement. What will be the result? We will see. I think this was the 2 comments I will to give. Actually, the question I want to ask Dr. Chatterjee, actually, because as the study published in New Jersey Medicine regarding the, I think in 2014, I think, regarding this Simplicity HTN3. This study is absolutely not acceptable, or it will not approved by ethical committee in China, because you using this ablation, this catheter to ablation the renal denervation, the other is a sham therapy, and this could be not approved by ethical committee because they will not let you do that. My question is, based on your AI help, could we in the future to using some calculation or some model to eliminate what we call sham therapy in the clinical practice or in the clinical trials? Because if in the clinical study using placebo-controlled, double-blind, maybe it's okay, but sometimes it's still difficult. But for sham therapy, it is very, very difficult if we design such a trial to get approved by the ethics committee. Dr. Chatterjee.
Yeah, it's interesting because we see significant country by country variation in the acceptance of different methodologies. So, what I found is that, the European Medicines Agency will take cues in certain ways that the FDA might be guiding, and maybe the other countries might follow suit. But it's not always the case. We have found, for example, the EMA has traditionally balked, for example, at single-arm study designs. They've always wanted, a randomized controlled trial. But then we recently saw an approval in an oncology indication that'll be a decision that's forthcoming in the next couple of weeks. where they explained why it was clinically acceptable for a single-arm trial design to be used in this case. And I think what we're finding in, with the Chinese authorities is that it's not a silver bullet by any means, right? To say that a synthetic control arm would work or that certain AI-based methods would work. There's still significant considerations to how you think about representative populations, the explainability of the methodology, and also it's very, very therapeutic area specific. All of these decisions cannot be applied in a blanketed way across different therapeutic areas. So I don't have a good answer for you because I think it's very case by case. What I have found is that it takes one to create a regulatory precedent, and then everybody can follow suit, right? And that's kind of what we're finding for, as an example, in the synthetic control arm space, there's a path forward now for recurrent glioblastoma in the United States because, a company received approval from FDA CDER and from FDA CBER, and then other companies are designing their hybrid designs for their trial similarly. So I would expect, companies can hopefully put forward something like this, within China. We've had a number of conversations with Chinese companies on this type of methodology. They're all a little bit, shy in terms of whether they want to put it in front of regulatory or not, though.
Yeah, Fei, please move on.
Sure. So our hold on. So I think you can see my screen, and we also have a commentator, Dr. Shengxian Tu. So his research, as you have already heard, in his research focuses in the development and clinical translation of intelligent technology for diagnosis and treatment of cardiovascular disease. And actually, his technology, QFR, assisted by AI, has been used in Dr. Qiang Zhao's clinical trial. And so, Dr. Tu, so please make some comments.
Thank you, Dr. Zhao, for the introduction. So I would also like to make some comment on the topic today. I think AI is very broad. So machine learning is one of the branch, and deep learning is a sort of sub-branch. And over the last decades, I think, last year, I think deep learning has gained considerable interest since it really outperformed conventional machine learning methods. So the enthusiasm has spread. And now it also comes to the clinical trial design. It's the topic we are discussing today. So in this context, I think that when many people are looking at the topic that we are discussing today, they might think that it's deep learning because it's just in the context. But actually, I think that in the field of the clinical trial design, I think predominantly we are still using the machine learning. So we are not yet go to the deep, say, using the power of the deep learning. So if we really want to use this tool to improve the clinical trial design and also reporting and synthesis of the trial, I think that the quality of the data is extremely important because deep learning is data-driven. And I think we probably need to really spend the efforts in standardize the acquisition of the data and reporting the data and sharing the data as well as public efforts. So this is my comment to this topic. And I would also like to make a response to Dr. Drazen about his comment on Professor Zhao's talk about the resistance of the subject to AI during patient enrollment. So actually the AI QFR is not actually not to replace the QFR. It's just to make the automation, to improve the automation. So the principle or the methodology has been extensively validated before, and now using the AI, you really make the computational speed, reduce the computational speed. So then the computational time has been reduced from like previously 5 minutes to 1 minute, and now can also be analyze the multiple branches at the same time. So that would make the technology more easy to use. And the ease of use is, I think, is very important to get the adoption of coronary physiology in this field. So, and then let make a small comment. So we will not make the resistance of the subject to adopt this technology. Thanks.
Thank you, Professor Tu. And now we'd like to hear from Mary Beth Hamel.
Thanks, Jeff. And thanks to the speakers for your excellent talks, which I really enjoyed. Randomized controlled trials, as we all know, they're very expensive. They're often very slow, and they're often not generalizable. I think the AI tools that I heard about today to assist recruitment and to improve trial design really have great potential and to make trials more efficient. And I expect that they will be welcomed by researchers, patients, and clinicians. What's more controversial is the idea that we can generate robust evidence to guide the care of patients without randomization. That's, quite controversial. And, I guess what I would say is that natural language processing and AI have certainly, really increased the power of analyses of electronic medical record data. But the inherent limitations, as someone who generates that data myself each week as I care for patients, the limitations really are the quality of the data, and then, of course, unmeasured confounding. And I guess, from my skeptical view, I would have a hard time basing too many of my clinical decisions on data that are not based on randomized trials. So I would be in favor of making trials, much more efficient and much more likely to be successful rather than replacing them with virtual trials or non-trials at all.
Okay, so for those of you who don't know, Eric Rubens had a power failure at his home. That is, the electricity is not working. So he's unable to, he's listening in on the phone, but he can't really communicate with the group because of the intermittent connection. He gives his group the best, and he thinks it's, from what he's been hearing, it's a very good conversation. But he will turn the floor over to Zach Kohane. Zach, you're on.
Great. Well, I found this to be a really useful discussion, and I want to thank the various presenters and panelists. And so I will only, first of all, echo that I do think that part of the advances that we're going to get in clinical trial performance don't actually come with AI, actually don't come from the AI itself, but from the impetus that AI will give us to learn and therefore gather data from other trials so that we actually improve our knowledge of a given trial based on other trials with similar or related drugs. And I think the previous panelists have made that point already. So let me try to make a different point. When you're conducting a trial, you want to make sure that you are comparing the right groups. And so, for example, if you are trying to distinguish the performance of a drug, let's say on a stage 3 cancer, it's going to be a muddier trial if some of your stage 3 patients are stage 2 patients and you don't know it. How might that happen? It might happen if you just use a standard pathologist. When we look at tissue, pathologists in the best academic centers will often agree with each other at best 85% of the time. In community hospitals, closer to 65% of the time. And I do believe that this is where machine learning and high performance on looking at, in this case, images of tissue and grading and staging the tissues will, in fact, create higher reproducibility and reliability of the class definition of people going in So, eliminating the low grade or the high grade, depending on what your study is, from the control group and from the intervention group, so that you actually have a much clearer signal. So I think that will actually improve the performance of our trial. So if you have an effective drug, for example, by clearing up that noise using AI in the way I just described, I think you will increase the likelihood of demonstrating an effect. That's number one. Well, that is my point. But it's not just in pathology. When we do neurological trials around, let's say, Parkinson's, we base these assessments on where you are in Parkinson's, on a clinical assessment. Even with a really great doctor, they're only seeing the patient for a few visits at most. Having a much better assessment of the progression of the disease, of the gait, of the speech patterns across even a month would be vastly more sensitive and specific as to the disease state in that neurodegenerative disease than the current assessments. So what I'm arguing is that the actual definition of the patient state, the phenotyping in a trial context, in a randomized controlled trial context, can be much improved. And that is as important in essentially pumping up the power of the study as recruiting more patients. So Dr. Drazen had pointed out that recruiting patients is important, but we don't have to recruit quite as many patients if our signal is less noisy. So I'll leave that as an area which I think is still underdeveloped in terms of the opportunity for AI. And maybe actually Dr. Chatterjee can actually tell us if industry is heading more in that direction.
Yeah, I think. Sorry, go ahead.
Arnaub, please answer.
Yes, so I think the idea is, I think there's a spectrum of how industry is receiving these types of technologies, how they are all pursuing data in different ways and what their plans are. But, I think the most innovative companies are deploying a number of different machine learning based methodologies. Some companies have publicized on this, pretty expansively, like the Janssen pharmaceutical companies, who are thinking about this in a number of different angles, where they're using, for example, imaging as an AI-based imaging tools as the basis for, trial submissions. They're putting in hybrid designs or hybrid controls as part of their submissions. So I think industry is getting there. It has become an education process. And I think most AI is not in the design phase. It is in the preclinical and the translational phase, and it's in the post-launch or in the commercialization phase. So that phase 1 to phase 3 window is still relatively untouched, in my opinion, or people have been scarred by previous experiences in terms of how they've thought about these methodologies and the regulatory side of it.
Okay, thank you. Adam had a question, please. Adam?
I was just going to respond to Zach's comments before as well. I mean, I think improving the quality of the data that goes into EHRs or into trials is obviously a big thing, and enriching EHRs as well as standardizing diagnostics is the scope in which many of the AI advances have been made already, and I think there's something like 400 AI methods that have been approved by the FDA for diagnostics. Most of them are in imaging, and that would, of course, improve the standardization and the quality of the data that can go into trials and or into EHRs. I think that. For example, NLP is also useful for enriching the quality of data that comes from EHRs by extracting more information from clinical notes, which goes to Mary Beth's point around, can we trust EHRs as a source of evidence? And I'm not going to pretend to be an expert in causal inference over EHRs because I don't think that's necessarily the right way to go. But we can embed prospective randomization blinding into EHRs if we're able to make sure that EHRs are capturing data as well as a clinical trial would. And when Arnaub was talking, I was looking at it and thinking, a lot of us are sort of like struggling. We have access maybe to Yoda and Vivli, and we don't have access to hundreds of thousands of patients over so many trials. And I was just thinking, wouldn't it be cool if the 2,500 CAR T patients were de-identified data that was released to the community as a shared task? And we could perhaps reduce our false negative rate down from 15% down to 5% by using something other than XGBoost. And there's lots of smart machine learning people around the world who could have taken advantage of those de-identified data if they were made more widely available to the community.
So thank you. So we have just a few minutes left, and I want to see if there are any other questions from the speakers. Dr. Zhu, we haven't heard from you after your presentation. Do you have any other questions or comments you want to make to the panel? So I guess, please.
Sorry, Dr. Zhao Qiang or Zhu Yunpeng, do you have any comments or questions? You're on mute right now.
I have a question for Dr. Dunn. Can machine learning give us how the importance of the evidence gap is? And the second question is the sensation can produce a guideline for real disease instead of or without a clinical trial?
I'll attempt to answer. So I think this also goes a little bit to can we compare the evidence that might be produced by a solution that's fully embedded in EHRs and uses retrospective cohort analyses with a prospective randomized controlled trial. And it's not quite clear to me how many times we've attempted to do both of those at the same time to see whether or not there is actually an evidence gap. And we know there are advantages and disadvantages for both. Obviously, intention to treat bias is a problem, but then generalization of the population is also a problem in the other end. So the question is, if we do the same thing at the same time, are we going to generate the same evidence base? And I don't know that the answer is there.
Well, Adam, I think you've picked up on one of the key points that we have. One of the key things, this is about aims in medicine. AI has great potential to actually change the way we do clinical trials. We've heard about their potential role in design, in thinking about what the best trial to do, about recruitment, enrollment, retention. Zach brought up the data that can be gathered. And I think that, there was a program about Oliver Sacks, the neurologist that used to go to visit his patients at their homes so we could see how they were doing. And what we really can do is, can AI give us a much richer data set with respect to a drug that is going to change people's behavior? So that is the data itself that are being gathered, the analysis of those data, and then the conclusions we draw from them. So we see great promise here, a difference that AI can make and will make, we think, in how we gather the evidence that keeps health professionals doing the job that we want to help them do, which is caring.